Good morning, everyone. Today I will be sharing with you about mesenchymal stem cells and their application in cell and gene therapy. I will begin by first introducing to you what is mesenchymal stem cell, followed by their therapeutic properties and their mechanism of action. I will then share about MSC-based therapy, genetically modified MSC, and end my talk with challenges of MSC-based therapy. A.J. Friedenstein, a Russian scientist, first discovered mesenchymal stem cells about 40 years ago when he made the critical observation that the bone marrow in postnatal life is a reservoir of stem cells for mesenchymal tissues. Friedenstein and his co-workers, in a series of seminal studies from the 1960s to the 1980s, isolated for the first time adherent fibroblast-like clonogenic cells from the bone marrow. The cells showed a high replicative capacity in vitro. In addition, the cells were able to differentiate into osteoblasts, chondrocytes, adipocytes, and hematopoietic supporting stroma from the progeny of a single bone marrow stromal cell that was retransplanted in vivo. The term mesenchymal stem cells was first coined by Arno Kaplan in 1991 to refer to the progeny of a small number of multipotent cells in the bone marrow involved in the formation of bone and cartilage in embryo and repair and turnover in adult. These MSC can divide and their progeny become committed to a specific phenotypic pathway resulting in end-stage phenotypes as illustrated on the figure on the right, including bone, cartilage, ligament, adipocyte, and dermis. Since then, MSC have been successfully isolated not only from the bone marrow but from a variety of tissues such as but not limited to adipose tissue, umbilical cord Wharton's jelly, umbilical cord blood, placenta, amniotic fluid, dental pulp, and synovial membrane. With the many different tissue sources and methods of isolation, the resulting MSC may exhibit different characteristics. In 2006, the International Society of Cell Therapy published a position paper on the minimal criteria to define human MSC to foster a more uniform characterization of MSC and to facilitate data exchange among researchers. Up until today, this set of criteria is still widely accepted and used to define MSC. The minimal criteria set by ISCT is, first, the MSC must be plastic adherent when maintained in standard culture conditions. Second, MSC must express a set of cell surface markers, namely CD105, CD73, and CD90. The MSC also lack expression of CD45, CD34, CD14 or CD11b, CD79A or CD19, and HLA-DR surface molecules. Lastly, the MSC must be able to differentiate into osteoblasts, adipocytes, and chondroblasts in vitro. MSC have distinct characteristics that make them attractive candidates in cell-based therapy for the treatment of human diseases. Firstly, they are easy to isolate and culture in the lab and produce in large scale for clinical usage. Secondly, the plasticity of MSC enable the differentiation into various cell types of the mesodermal lineage. MSC also have homing ability to the site of tissue or organ damage while exhibiting little or low immunogenicity due to the lack of expression of the co-stimulatory molecules. Lastly, MSC also secrete bioactive substances such as growth factors and cytokines in addition to extracellular vesicle that regulate the local cellular responses to injury. The therapeutic potential of MSC is due to their two main specific properties, which are the abilities to home to the site of injury and repair the injured tissues. Tissue repair is achieved through the secretion of various mediators such as immunomodulatory, anti-inflammatory, anti-apoptotic, antimicrobial, antifibrotic, and angiogenic agents. These mediators are also involved in the activation of resident stem cell population in the injured tissue to stimulate endogenous population of stem cell proliferation. Homing refers to the selective migration of MSC to the site of injury and followed by sustained delivery of the trophic signals for tissue repair and regeneration. The migration is in response to the expression of specific receptors and ligands by the injured tissue or gradient created by chemoattractants due to accumulated chemokines and cytokines in the tissue microenvironment. This homing ability is highly useful when the sites of injury are spreading or not easily accessible for delivery of treatment. The figure here showed the homing mechanisms of MSC. MSC express CD44, which binds to selectins expressed by endothelial cells to facilitate the rolling of MSC along the vasculature wall. This is followed by activation step upon the detection of inflammatory signal, which will cause the MSC to be arrested and begin to transmigrate through the endothelial cell layer and basement membrane. Lastly, MSC will migrate through the interstitium to the site of injury based on the chemotactic signals released by the injured tissue. Homing by MSC can be achieved either through non-systemic homing or systemic homing. In non-systemic homing, it is done through local transplantation of MSC at close proximity to the site of injury and may require more invasive procedures. Systemic homing, on the other hand, is done through intravasal delivery of MSC. This method, while less invasive, requires the targeted extravasation of the circulating MSC at the site of injury. When MSC reach the site of injury, they contribute to tissue repair and regeneration through the secretion of various mediators as mentioned in my earlier slide. This repair ability is further enhanced by the ability of MSC to exert pleiotropic effects. Among the many pleiotropic effects, anti-inflammatory and immunomodulatory properties are widely believed to be responsible for the therapeutic benefits of MSC. MSC secrete factors such as TGF-beta, TNF-alpha, and interferon gamma that will interfere with the immune response and subsequently modify the inflammatory landscape. This in turn confers protection to host cells by reducing the severity of immune response to inflammation. At the same time, the modulation of immune system by MSC also activates essential inflammatory processes responsible for healing, scarring, and fibrosis to promote tissue repair and regeneration. This slide is again to illustrate to you the various mediators secreted by MSC to achieve tissue repair. These mediators activate the homing activity and exert various pleiotropic effects as shown in the slide. Now I will move to the mechanism of action by MSC. In the early days, the therapeutic applications of MSC focused largely on the multipotent differentiation ability of the cells. MSC then were viewed as potential cell source that upon engraftment after transplantation will differentiate to replace the injured tissues. However, over time, the focus shifted more onto the ability of MSC to secrete bioactive substances such as growth factors, cytokines, and extracellular vesicles. This mixture of product is known as the secretome of MSC. MSC secretome exerts its healing effects through paracrine signaling to stimulate innate tissue repair and modulate inflammation and immune responses. There are currently 328 active and recruiting trials involving mesenchymal stem cells or mesenchymal stromal cells as keywords in the database of clinicaltrials.gov. Most of these trials are conducted in the United States, about 102 of them, and China, about 95 of them. Of these ongoing studies, the majority are currently in phase 1 followed by phase 2 trials. There are also 329 completed trials involving the same keywords in the database. The trials covered a broad spectrum of diseases with the majority on autoimmune, cardiovascular, and neurodegenerative diseases. Other prominent diseases include renal and liver diseases, diabetes, and cancer. Autologous and allogenic MSC have been used in cell-based therapies to repair and replace damaged tissues and enhance the functions of tissues and organs or to exert immunomodulation. In terms of the route of administration in cell-based therapy, as you can see from the diagram, MSC after clinical expansion can be transplanted into the site of injury, or their secretum can be collected for systemic infusion. As mentioned earlier, MSC have been used in broad spectrum of diseases. Now I would like to take a moment to highlight some of the prominent outcomes of MSC-based therapy. I will first cover the outcomes from transplantation of MSC. The first one is a graft versus host disease trial on 301 patients conducted in 2015. The patients were treated with IV infusion of related and unrelated human MSC from matched and mismatched donors bi-weekly for 4 weeks. Results showed 136 patients with complete response and 69 patients with partial or mixed response. In total, 205 patients showed overall response. The second is a phase 1-2 trial on refractory systemic lupus erythematosus that was conducted in 2014. 40 patients were given IV injection of allogenic umbilical cord MSC. The outcome of the trial is satisfactory clinical response. In the hereditary cerebellar ataxia trial, 100 patients were treated with allogenic umbilical cord MSC transplantation in the subarachnoid space. The patients showed significantly improved multiple neurological functions. The last highlight is a phase 2-3 trial on acute myocardial infarction. The trial was conducted on 80 patients using intracoronary administration of autologous bone marrow MSC. Outcomes showed tolerable and safe with modest improvement in the left ventricular ejection fraction of the patients. Now I will highlight some findings using MSC secretome in preclinical studies. The first study on lung disease, the extracellular vesicles from human bone marrow MSC resulted in the attenuation of oxidant-mediated injury in the alveolar type 2 cells. The second study on acute liver failure used the conditioned medium and exosome from human bone marrow MSC. The results showed inhibition of apoptosis and enhanced proliferation in the hepatocytes. The third study on inflammatory bowel disease used extracellular vesicles from umbilical cord MSC. The results showed reduced production of inflammatory cytokines by macrophages. And lastly, in osteoarthritis model, exosome from bone marrow MSC reduced apoptosis and enhanced proliferation of chondrocytes for cartilage regeneration. MSC have also been genetically modified to express therapeutic genes. This modified MSC can be utilized for the targeted delivery of therapeutic gene product as gene therapy. This method will overcome the problems of direct administration of therapeutic genes, such as the short half-life in the body, and also toxicity associated with high dosage administration. The use of MSC for delivery of the therapeutic agents can help to minimize such problems because MSC can selectively home to injury site and exert therapeutic effects locally to increase the concentration of the agent for better efficacy while at the same time reduce the systemic toxicity. Genetically modified MSC may be utilized for treating a range of genetic or acquired diseases as well as cardiogenic disorder, neurological diseases, and even cancer. This figure illustrates the usage of genetically modified MSC to deliver therapeutic genes. MSC can be isolated from patient or donor and genetically manipulated in the lab to express a particular therapeutic gene. The modified MSC will then be expanded in the lab prior to re-infusion of the cells into the patient. For the last part of my talk, I would like to highlight some of the challenges of utilizing MSC-based therapies in clinical applications. The first challenge is on the heterogeneity of the MSC product, which depends on the isolation and culture methods in addition to donor factors such as the age, genetic traits, and medical history of the donor. Secondly, the administration of exogenous MSC may also have side effects, for example, formation of undesirable tissue, as the MSC may predominantly give rise to bone and cartilage cells. The administered MSC may also have short survival to confer any therapeutic benefits to the injured cell upon transplantation. Lastly, MSC may also exhibit pro-tumor activity due to their pleiotropic and immune suppressive effects. Despite these challenges, the advantages of MSC in clinical applications make these cells an important medical product for further research and development for cell-based therapies in the future. Before I end my talk, I would like to summarize again the advantages of MSC for clinical application. MSC can be easily isolated from several sources and produced in large scale for clinical usage. They can also differentiate into a variety of cell types and exert pleiotropic effects, making them attractive candidates in the treatment of different pathological conditions. This is the list of references in my talk. With that, I thank you for your kind attention.